And I'm going to talk to you more or less about the methods that we have. I know the title I gave is fairly convoluted, although pretty accurate. I'm going to talk to you about the imaging that we do at the beamline and how you can benefit best from that. So I'll go straight into it. And then I'll just introduce, you will know that Diamond Light Source is the UK synchrotron. I have it down here. And within it, quite a few groups. Our beamline is part of the biological cryoimaging group. And we have a website here. I've put down the website also. So please tap into our website for any information. And I have the photos of all of us and all the lovely people that contribute and work at B24, our students, scientific advisors, all of the people who are likely to meet if you were to visit the beamline. So I'll start by saying what I'm going to be talking about is methods that will help visualize up to the range of 225 nanometer resolution. So that positions us very well to looking into cells, organelles within cells, some of the higher micromolecular complexes, and perhaps thin tissue slices. So I will start with an absolutely dreaded kind of slide where there's a lot of words, and I kind of hate those slides when they show up, but they are actually necessary. So for your benefit, I've put a little cartoon here just to distract you a little bit. But I will start by trying to pass on who can benefit from this method. So I will start by saying, first of all, what are the projects? What is the right project focus? So who could use B24? In general, the person that the group that will use B24 will have a project focus studying cellular ultrastructure and chemical localization within it under a known set of conditions. And this is a fancy way of saying that is you're probably looking into cellular mechanisms, maybe studying the effects of drugs on cellular structure. And when I say drugs, it could be anti-cancer compounds, it could be vaccines or similar. You might be looking into the steady state or induced organelle and substructure morphology and distribution within a cell, looking into infection mechanisms at the cellular level, or perhaps even pathogen defense and clearance mechanisms, development and differentiation, and many more. So that gives you an idea of the type of projects that we see at the beamline. Then these projects need to require our method. So our method needs to be the right method for that project. So in general is whenever microscopy data is required, but with some conditions. So the project need to be able to benefit from snapshot imaging. As I'm going to mention in a little while, the samples that we look at, we fix them through plunge freezing, through vitrification. So we freeze them very rapidly without destroying any of the fragile cellular structures. But that means that we cannot witness dynamic processes bar snapshotting, freezing at specific times. So if the project requires dynamic live imaging, certainly we cannot help with that. The other thing is that 3D data needs to be required. So you need to be able to have an intrinsic requirement for your data to be represented in 3D. Now, cells are not two-dimensional. They're three-dimensional, so that's well suited. But if 2D data is required, then I can recommend a number of other methods that will give you higher resolution, but you lose the third dimension. And then you need to be looking into single cells and sometimes cell populations. And you need to understand also that cells, even within a monoculture, perhaps they came from the one progenitor and they're experiencing the same environment, have personalities. So organelles and features and structures within cells are not like molecules in a crystalline lattice. They actually differ from one to the other, which is why I said personalities. So if you're looking at the subtle effect that a specific drug has caused, you really have to explore the steady state control population to figure out what is outside that range of personalities. So it is in some ways, it requires a commitment, to explore this. And it needs to be in the right time of the project. So the method can be used at an initial stage when people are being looking at a project or an area in an explorative fashion. And that's okay, but it's a high-risk method in that what you get out of it might not be the cornerstone of a big project. Now, when you have a mature project with well-defined expectations, that's what we really like to see, because then we know what to hone in onto. And then if correlative microscopy is what exactly is needed, then if the reagents like fluorescently tagged proteins or similar are around, even better. That's really exciting then. So apologies for the long slide. I believe it's quite essential to give you an understanding of what we can offer. So this is it. All right, so I'll go into what we do as a team, the lovely people that I showed you before. We design and build X-ray invisible light microscopes, fundamentally. So we develop what we like to consider user-friendly, but certainly cutting-edge microscopy platforms. And we're here to help from the very beginning. We can help with designing experiments, protocols, expertise, data collection, data imaging, data processing, and analysis. We all have our individual research projects, so we're not that far apart from our users. And I have a few snapshots of data that we've collected, but we'll see all these in detail in a little bit, and also the correlative workflow, which is the next thing I'm going to show you. Okay, so at the beamline, we examine cells on TM grids, and I will go into this a little bit in the next slide. These cells, these samples, when they're ready, they enter a correlative microscopy workflow. They can go into conventional fluorescence, super resolution fluorescence microscopy, or X-ray microscopy, tomography. And when we're done, the same sample can go into other modalities, depending on user needs. The essential thing to take home from this is that it's the same sample that can go through all of these microscopes. Therefore, the correlation of data becomes unambiguous. And I have snapshots of our X-ray microscope and a fluorescence microscope, which is in fact right behind me also. And an example, a graphical example of the type of data. Now this I will talk a little bit more, so I'm not going to focus on this right now, but you will see it again. And one last setting the scene slide, which is rather important. Quite often when I talk about our method, I talk about a sample being captured and imaged in near physiological states. And this brings about a lot of debate and a lot of questions. Why I'm saying this, and I'm going to explain what this means. We have a question between the required versus the necessary geometry. The required architecture, how you would like to see cells, are within context. So a cell is part of a tissue, maybe a population is three-dimensional. It interweaves between matrices, and this is just looking at just the epithelial layers within skin. So as an example. So this is ideally how you would like to have your cell when you're looking inside it. This is clearly not possible at high resolution. So what we do is we opt for a necessary geometry, which allows us to image at high resolution at near physiological states. So what we do effectively is we take these cells, and either we let them grow or we deposit them on an electron microscopy grid that has some sort of a carbon surface for support. So we don't flatten physically the cell, but we allow it to settle. That is called, in effect, is a flat geometry for imaging purposes for some people. But I would like to point out that it's always three-dimensional. The cells, when we look at them, have a depth of, depending on their lineage, maybe 3, 4, 5 microns. To some fibroblasts that are like literally 10 microns thick. So it's not that we're missing the second dimension, but I will quite often mention flat geometry because we're using a flat surface to grow them on. And I'm showing here a couple of TM grids. We use gold grids to be amenable with tissue culture. And these are on a cover slide, and that's a comparison with a 5p coin. So pretty small, pretty fragile. We use any number of grids, and these are necessarily matrices. We prefer to have finder grids, as you see here, and that helps us to position when we move from one microscope to the other. And all of them, as I said, have a support film, carbon support film, where the cells rest. Now, this support film could look like this, which is lacy carbon, or could look like this, like this is a specific type, which is called quantifoil. You'll be seeing it again and again in my data, so I'm pointing it out now. It's the support film where cells rest on. Okay, so at the beamline, we have our own workflow. It's fully commissioned, and you'll notice there's several stages. We take from cell culture, live cell imaging, and all the associated sort of work to plunge freezing, and then we go into microscopy, super resolution fluorescence or X-ray, and then we have an in silico side. One thing that I've highlighted here is the cryoimaging, because once the samples are ready to be plunge frozen, we move from whatever the growth conditions are. 11 to 37 degrees, I believe, are sort of the average. We have some extremes depending on the system. Your samples move then into liquid nitrogen, -1, -6 degrees. and they stay at liquid nitrogen. So we don't thaw the samples, they remain in that condition. And while in that state, they can travel through microscopes. And then, of course, the in silico work we do at room temperature. Depending on the lab that you are, these could be polar freezing or very, very hot. So, all right, so the only other thing that I wanted to say is that people can access this workflow at any point. There's no requirement to start at cell culture with us. It could be that somebody prepares their samples, does their cell culture, maybe they have experienced facilities to do their plunge freezing, and then send it here. Some people just access the cryosim, for example. Some people just access the X-ray microscope. Some people just get in touch with us to give them protocols and help with the processing. So users can interface with this workflow at any point. And I will start. So what I'm going to do is I'm going to highlight different sections of this as I'm showing you what we do at the beamline. I'm going to start with the plunge freezing because it's super important. So one of the most crucial things is the preparation of samples. And for us, because it's cryoimaging, the vitrification process is very, very important. I'm showing here an autogrid. So this is a finder grid where you can see that each position is marked by a letter number. And in it, you can see little cells that are growing on top of the support film. Now, these are G2S cells and from one of our projects. And you notice that we have a fair distribution. They have company, so you don't have a single cell anyway. They're nice extended geometry, some of them, and then some of them have fluorescence also. So this is the same area in fluorescence. It is absolutely vital that the preparation of the sample is done in a good way, because something like this, for example, where you see in the middle of the three a very good distribution is likely to give you good data. Whereas if it's crowded, like the view on the far right, it's unlikely you collect good data. You got a lot of noise, X-rays will be attenuated. And then if your cells are too sparse, like the one on the left, then it's quite likely that you won't be able to, they won't withstand the blotting because we actually remove some of the liquid, all the process of vitrification. So again, we can help with protocols for all of this, but it is essential that people understand that this is one of the most crucial things in the whole preparation. We cannot give you good data out of bad samples. So you have to start from a good sample. And I've stepped over the mapping. So once samples are prepared, we prefer our users to map them in a standard cryo-imaging setting to make sure to provide us with evidence that they're good samples. But the most important thing for us, and usually when we see a lot of samples coming in, is for the super resolution fluorescence imaging. Now, fluorescence imaging will have to always precedes X-ray imaging because we don't want to damage the fluorophores. So at the beamline, we have a cryosim. There's a cryostructural illumination microscope. It also has a D-storm capacity. And this was built by Mick with Ian also. Mick has moved on now, but Ian, for his sins, is still around and helps us all the time. So we're very fortunate to have developers working with us. So the cryostructural illumination microscope will accept vitrified samples on grids. It has the full range of excitation energies. We have 4 lasers, so any fluorophore we can excite to some degree at least. We can do geofluorescence register at the same time. It's 3-dimensional imaging, so it's Z-stats. And we can go through depths of up to 12 microns, in fact, more, but then you just start having reflections after that. And then a resolution to better than 200 nanometers, depending on the wavelength of your fluorescence. And that's fully commissioned. So this is fully accessible there for people to use. The cryo-D-storm facility is under commissioning, I'm afraid, because we're still having issues with fluorophores are just beasts at liquid nitrogen temperatures, and they just won't blink that easily. So we're still working on that. And just to prove a point, I can show you a 175 nanometer bead and how it looks in diffraction limited imaging on a microscope and by applying structural illumination. And the same with a cell that you will see actually in a little bit also. I'm using it as an example. In here, you see vesicles that are fluorescent red. These are actually endosomes of a single cell. The cell is here. The nucleus is in the middle where we have absence of fluorescence, and this is diffraction limited imaging and the SIM processing, the structural illumination processing. Now, all this system and its applications, as well as how to build it in some way, is also documented in our Optica paper. All right, so I'll give you a flavor of how this experiment happens. This I've taken from a project of a group that was studying cytoskeletal scaffolding proteins, and they were using fluorescent F-actin fluorescent U2S cells. So these are cells that have filamentous actin that has red fluorophore on. So when we do a sim experiment, you start by doing a Z-stack, and this is literally taking slices by moving the sample through the focus position. And I'll do it one more time. You go through the sample and we've reached the support film with the holes there. Now, cells are not extremely photogenic with white light. They look very, very transparent. So not a lot there, but the same area when we collect data looks a little bit like this. So this is actual raw data collection. And what you're looking at is the structural illumination pattern interfering with the fluorescent centers in the sample. Now, this happens in 3 angles. So you notice the lines will change direction now, and it's there in the second angle. And 5 phases, we translate them, hence it looks like it's moving. During the reconstruction process in Fourier space, the constructive interference between the known pattern and the unknown pattern is used to extrapolate features beyond the diffraction limit. So how this looks, let me go back in. This is the same data, but that's diffraction limited now. So if there isn't any structural illumination, that's how it looks. And if we go from the very top of the cell and slice through it, this is more or less our red signal within the cell. Again, very, very obvious where the fluorescent signal is. However, once it's processed, this data becomes a lot clearer. And you'll see as we're moving towards the bottom of the cell, the fluorescent signal becomes distinct, and we can get all that information from the actin cytoskeleton component. All right, so this is structural illumination. And as I was saying, you can use this to go from conventional to super resolution, and then you can explore your information in 3 dimensions. So in this case, that group was looking for the presence of specific proteins on this scaffold. And they were testing it in both interface and mitosis with a characteristic cell shape. But I'm only using it here as an example of what you can see. Okay, so this is the fluorescence microscope. As I said, the other high-end microscope we have is a Crysoft X-ray tomography microscope. Now, I won't go into the theory too much because I'm aware there's another webinar of the same series that actually went into the theory a lot. But I will introduce B24. This is our cabin. The microscope is at the far end here on the left. And then if you turn around 180 degrees, this is the wet lab area of the beamline. And in fact, this is super busy right now because we have several tissue culture hoods now in that area, and it's our wet lab environment. This is us working hard for the purposes of a photo session. And then the beamline, effectively, the star of the beamline is the microscope. Now, the microscope is a commercial system that's adapted for the beamline, and it has the standard optics of any microscope. It has a condenser, it has a zone plate, only their software soft X-ray adapted. So we have a capillary condenser, a sample position, and a zone plate objective, and a detector here. We take light from a bending magnet from the synchrotron, and we have a single focusing toroidal mirror. So in a nutshell, this is our beamline. And a little bit about the technique. As I said, I won't go into the details, but it is a 3-dimensional imaging technique. It allows us to look into the ultrastructure of vitrified cells at near physiological states, as I said before. It requires no fixation or staining or slicing or any other manipulation of the sample beyond the immobilization by plunge freezing. We use, the good thing why this is because we use the natural absorption contrast we get in a specific range of X-rays between the absorption edges of carbon and oxygen, at which point carbon-rich biological material absorbs X-rays a lot heavier, a lot more than the oxygen-rich media that surrounds it. So we get a natural absorption. And we have both 25 and 40 nanometers option for the resolution. Now, I need to say here that everything I've presented here is actually the work of an awful lot of people, but especially in this case, special thanks is to Liz, who advocated and saw the project take off, to Jeff, who is always there whenever we call him to help us with the technical sides, and Eva, who stepped in at the key points when we're having a lot of problems and helped us get through them. So it's very much a collective project. Now, I'm showing you here to the right, a video from the project of one of my students, Kamal, who's working studying herpes virus. Now, what you're looking at as two cells, the interface is in the middle, and on the left, you have a nucleus. On the right, you have another nucleus, and inside the nucleus, you can see herpes virus capsids. So the little spots that you see are in the region of 1,000 angstroms. Quite interesting. Kamal was looking at the egress process, so nuclear egress. So he was interested in the ultrastructure in the vicinity, perinuclear area. And you can see quite a lot of information, mostly to show you what the technique can do. The X-ray environment, I think, deserves a mention because soft X-rays don't travel well through air. So the whole microscope is effectively under a high vacuum environment. Mechanically, that is not an easy thing. And this is the working area where you see the optics, which we can't see them. Actually, it's very difficult to see them, but the objective lies here. And then we have a capillary condenser here, the sample in the middle. Anything goldeny is for cryoprotection. The whole system where the sample is cooled via conduction, because of course we can't have liquid nitrogen in vacuum. This is the direction of the beam, and I wanted to point out that we're fortunate enough in this system from Zeiss, which used to be X-Radia, this specific company. We can load 4 samples at a time. One of the trickiest things when you have a system that operates under high vacuum is transferring samples in and out. So having the capability of loading 4 samples at a time saves us an awful lot of time. And I will own up to the fact that this is quite speeded up. Okay, it's actually much slower, but at least 4 of them are there at the same time. All right, I'm going to skip this process because I'm hoping all of you will come to the beamline and collect data at some point so you'll see it in person. And I'll show you a little bit, one more example. These are U2S cells. And what you're seeing here is a 2-dimensional 2D X-ray mosaic where we collected one image. And these boxes that you see here are in fact one field of view each, and then move the sample a little bit, collect another image, move it a little bit. So we do a mosaic. So you're looking at one cell here and it has 3 little friends, one here, another one here, and another one there. And we collected data in this area specifically. In fact, we collected data all over the place, but this is the one I chose to show you. And our raw data is projections. We collect projections. What we do is we take the sample, we rotate it a little bit, take a projection, rotate it a little bit, take another projection. And then we combine all of these projections into a 3-dimensional volume. So this is the actual raw data as it comes from the microscope. So each time is small rotation, snapshots, small rotation, projection. And what you're seeing here, the nucleus of the cell is here. You see a couple larger lipid droplets. The little black spots are gold nanoparticles that we use to align each image with respect to the previous and the next. And then quite a lot of information within the body. And of course, it's already pretty impressive, and to some extent, the data collection can be quite quick. So if you're doing these exposures, for example, were only like a few seconds each. I'm lying, that was 1 second each. So you can imagine if we're collecting 120, 240, maybe 600 images, going through that, it could be 5 minutes to maybe 20 minutes, but no more per tomogram, per package of data. Now, when this is processed, of course, we enhance our signal massively because we record our signal on each one of our projection. And then when it's reconstructed, it becomes much more clear. And I'll show you this movie taking you from the top of the cell through the cytoplasm and all the way down to the support film. So to me, it's a really beautiful data. Of course, it would be. If you see now, we've just reached the support film and the movie will take us back out. In fact, perhaps we won't give it a chance. I'll return to a nice picture here. Well, you can see very clearly mitochondria, endosomes. In fact, if I'm not mistaken, exactly, I've reproduced it here. So in any snapshot, we can stop and figure out the different components. Here, for example, you see the nucleus, mitochondria, beautiful cristae, the endoplasmic reticulum wrapping around all the structures, endosomes, multivesicular bodies. So depending on the project, this is the kind of information you can harvest. As I said, at the beamline, we have 2 modes for resolution. We have 25 nanometers and 40 nanometers. Of course, everybody would go for the higher resolution, but you do get that at a cost in your field of view, so you get to see less. So for some projects, it's actually more suitable to use the 40 nanometer resolution. And just to prove a point, I can show you, to just show you the capacity, two data sets. This was collected with a 40 nanometer objective. This is with a 25. Both of these are cells infected with reovirus. And here you're looking at the interface between two cells. This is a data set you'll see again in a little bit. Whereas in this one, and this is early stages of infection, whereas this one is later stage of infection, and you have a cytoplasm full of viral factories effectively. Now, I like this one because if you zoom in on a slice and then zoom in some of these viruses, the same group the year before had published this paper showing the same, pretty much the same structures. And you can see the resolution of individual viruses here and what we see. So it's not a million miles away. But then the postdoc that did this took us, to my understanding, a substantial amount of time to do it, whereas we collected this data in literally 5 minutes. So I'm not sure he was that excited with our progress. So this is one of the benefits of the technique. You can actually capture a lot of information quite quickly. Okay, so bringing the two together. So I described the cryosim and described the cryo-60, wonderful techniques in themselves, but if you put them both together, you get something altogether much more amazing. So I'm going to use an example that we published recently to explain how these things are brought together and the type of extra dimension that you can add to a project like that. And in this paper, we were looking into reovirus infection. Now, reovirus is a relatively benign virus. It's a model system. It's got some pretty nasty cousins, but in itself is not a dangerous pathogen. And when it infects cells, it gets taken into the cells through most likely a clathrin-dependent endocytosis event. Now, that means that the virus finds itself in the cytoplasm. Tick, it's exactly what it wants, but unfortunately is sequestered by the membrane of the endosome. So it's not actually in the cytoplasm to actually go ahead with RNA synthesis. At some point in the early points of infection, something happens to this endosome, and we know that capsids cores get released into the cytoplasm. But this is an event that was rather obscure. There was in vitro data that this pores can be formed with one of the main proteins from the virus, but nothing in a real life system. So we were exploring this to see what's happening. And we used our methods through bringing together all the microscopies to get a three-dimensional correlative. And this is just a schematic that I have on the right here. It's also in the paper, but it will become more obvious as I go through in the next slides. So starting from the beginning, as I said, users can interact with us at any stage, and this specific group sent us grids already vitrified, already plunge frozen, very well prepared. These are finder grids, and they were loads of cells on each grid. All of the cells were infected. They used fairly high MOIs, fairly high viral titer. So we're fairly confident that all of the cells were infected, but only a very few of those cells were expressing a reporter molecule, which was an indication to us that endosomes became leaky. Hence, the virus somehow made them leaky. So here you see the bright field, just white light imaging, fluorescence, and I'm using the reporter molecule for fluorescence. In this case, in this box, you can see the single cell that is contains this fluorescent marker. And if it reminds you something, that's the one I used to show on the cryosim page. So what we did was, in effect, take snapshots. As I warned at the beginning, that's how we follow dynamic processes. We take snapshots. So we use samples that were mock infected, 1 hour post-infection, 2 hours post-infection, 4 hours post-infection. And in this process, we were able to, just by using the cryosim, follow the accumulation of viral components. The viruses were all green in vesicles that were also fluorescent label, which means that we had become leaky by this stage. So we believe that by 2 hours, the virus had managed to inject its core, release its core into the cytoplasm, but causing the vesicles to become leaky in the process. So a lot of their supporter molecules seeped in. It's quite impressive, though, that all the vesicles look structurally intact. So it wasn't like they exploded or anything. They literally just became slightly leaky. And then by 4 hours, we find the virus and this reporter molecule concentrated in larger vesicles. So we already had an understanding about the timings. And then to correlate that data with the X-rays, we collected X-ray data. And this is an X-ray mosaic, as I said before, with several cells. What we did is in silico, we brought the bright field data first from the SIM, so just white light data from the microscope, the fluorescence microscope, onto the corresponding area. And that allows us to bring the fluorescent imaging of the same area into the correct orientation with respect to the X-ray data. So this is 2D alignment because remember the X-ray mosaic is 2D. Now that we have the fluorescent data in the right orientation, we can take the data that we collected in X-rays. So we collected 2 areas here, and you can see them here, and we're next to a nucleus here, and with quite a few vesicles and pretty substantial, respectable multivesicular body / late endosome. Now, if we focus on this, you can see the outside membrane, quite a lot of wrapping of membranes and other components here. It was red, so the correlation allows us to immediately point it as a leaky vesicle, so its membrane must have at some point opened up. And then we can clearly say that all the internal structures that we see are viral related. It could be viruses that were not perfect and did not release their cause, or other components of the original infection. So it immediately becomes obvious which vesicles are involved in the whole clearance, infection and clearance process. Which means that once we have, and I've gone back to the 2 hours now just to show you the data set of the movie, which we'll see in a little bit also. But if we have this extremely detailed ultrastructural, 3-dimensional ultrastructural map where we can see nucleus, bodies, endosomes, a lot of structures that we haven't seen before in these cells. And then once I put the fluorescence in, and I'm using only the green fluorescence, I can tell you immediately that this structure that I see here, quite heavy in carbon, remember the contrast we're looking is carbon here, is all viral related. I can focus on only these vesicles. I don't need to look at the others that are not playing any role. And of course, this whole thing is 3-dimensional, as you see here. And again, the same area, just to show you the comparison. This is just the X-ray by itself. Again, this is the data you've seen already. You're going to be an expert in this data set soon. And then the same thing, but now with all the fluorescence imposed on it, which means to me that I can go and get all my statistics on those specific vesicles so that I can study how the virus travels within the cell. Okay, so now that I've shown you what this can do, I'm actually going to take a step back, because it would be disingenuous of me to suggest that this is an easy process. Bringing the data together, 3-dimensional imaging data, is really stupidly hard at times, but not if the samples are prepared in a clever way. So for us, it's paramount importance if people are to benefit from the combination of methods, and also in a meaningful way be able to do this within the lifespan of a human, within the lifespan of a PhD or a PDRA, then you really have to think about how you prepare the samples. Well-prepared samples means that the data could be correlated semi-automatically, and you can do it in a day as opposed to spending several days per tomogram. So if you, I'm showing the super resolution here, fluorescence, and how these are brought together. So it's very important, first of all, for the samples to be prepared with correlation in mind. So you need to consider fluorescence of vesicles that you can track, as well as including fiducials like alignment markers that can be seen in both methods. So fluorescent nanoparticles are ideal, for example. We spend a lot of time building our experiments so the user at the end can have something that can easily be translated into actual real science results. Then also, if you're looking at the plunge freezing, super important. I mentioned this before, so I'm only saying it as an aside, that there's many ways to prepare a grid, and God knows we've seen them all, only a few that we absolutely spot on. So we are here to provide protocols, advice, and expertise. I hope people will take advantage of that as much as possible. Okay, so provided this has been taken into account when you start with the sample, it's likely that the process will give you results very easily and efficiently. So in our effort to make sure that people know what we're doing, we try to publish everything in our website, first of all, and I have it here at the top. But we try to literally publish everything we can. So we have publication, sample preparation, data collection, data correlation, data deposition and dissemination. We're particularly proud of this because this is one area that people are not thinking a lot. Once you have reported, or you need to report your results, your data needs to be deposited for evaluation by other scientists. So we have a paper now, which is going to be out on the 22nd of this year, on how to deposit the data, instrumentation and software development, as well as applications. So this is all out there. If it's not been published yet, we can certainly provide it. And in the same spirit, we have, yes, so to access all this, so it's lovely, how do we get about actually using it? All of this is documented again on our website. The Beamline's access through proposals, either standard access or rapid access, and clearly through our collaborations with us. So standard access involves putting in a proposal, it goes through peer review, and then it's allocated time. And with time comes us also, because we help with sample loading and data collection. And then it's allocated specific shifts or time on the instruments depending on their scoring. And then there's rapid access, which is much faster. It gives smaller times, but it's there for feasibility experiments. Now, currently, we're only taking rapid access because of COVID restrictions, and the access for our users is only remote. But I believe we're going to have a new call for standard access from March. So you find all that information here. Normally, there's 2 calls for proposals per year. And we also have ourselves. So for documentation and procedures, you're very welcome to give any of us a shout. Now, Ian is not officially part of our group, but bless him, he's just sort of like chained to the microscope, so he's also available. I've put my contact details, but you really can find all of our information here, and any of these lovely people would be very glad to hear from you. Okay, so literally the last slide. Acknowledgements, loads and loads of people. I mean, this was not the effort of one person, but many past and present team members, loads of people at Diamond. I'm underselling it here, a lot of people. We got help from the international synchrotron community, especially from Mistral at Alba, and from imaging, instrumentation and software development experts. Micron at the University of Oxford have been wonderful, and the X-ray imaging team at ZEISS has been also wonderful. Special thanks to the users who's completely unauthorized I've used images from in this presentation. But before I say anything else, the most important thing is that we are actually pushed and helped by user community. So people that come here to do their projects actually contribute in the development of the Beamline, and we're really super appreciative for that. So there's special thanks to them. Before I say thank you for listening, and that it definitely deserves a big thank you, we have a Twitter feed. So please come by at B24Light and say hello. And that's it. Thank you for listening.